All right, so maybe I'm going to start so that we keep on schedule. So at Sensorion, we're passionate about the hearing sense. Did you know that there are 1.5 billion people that are suffering from hearing loss? And this is data from WHO. And actually, there are different causes for hearing loss. 50% is genetic cause, then there's environment, drug-induced, or we don't know, it's idiopathic. And so the vision at Sensorion, it's to help people with inner ear disease disorders so that they can live their life with unlimited connection. This is my disclaimer. I'm Valérie Salanté. I'm Director of Regulatory Affairs at Sensorion. And I thank ARM for the invitation for me to present on behalf of my CEO, Nawal Ouzren, the therapeutic option that Sensorion propose to prevent, treat hearing loss, or restore hearing. I will start with a rapid overview of the company. Then I will focus a little bit on something that is core to Sensorion strategy. It's to build bridges with a strategic partner in the field of hearing. And then I will focus on our 2 gene therapy program. So currently, we are developing therapeutic options with 2 types of molecules. We have the first asset is 2 gene therapy programs targeting monogenic recessive forms of deafness in pediatric or adult onset. The first more advanced molecule is Sens 501, it's clinical stage, and the second one is GJB2. And for that, we have IND-enabling studies starting. And for those 2 programs, there are natural history studies ongoing to support the programs. On the building of a franchise on hearing loss, we also have 1 small molecule that is clinical stage and that is developed in 3 indications. So preservation of residual hearing after a cochlear implant, ototoxicity induced by cisplatin, and sudden sensorineural hearing loss. For our program, we have an exclusive relationship with Institut Pasteur in the field of hearing genetics, and also partnership with the hearing care and medtech players. Then, in terms of financing, we're listed on the Euronext market growth, and the slide is not up to date, but we were able to raise 3 times this year in about 9 months for a total of 100 million, and there was a press release yesterday that we raised additional 15 million. Now I will tell you more about our partnership that covers translational research, clinical research, and also in diagnosis and the medtech. So our gene therapy journey started back in 2019 with a very important collaboration with Institut Pasteur. Institut Pasteur is a renowned academic institution, and there the team of Professor Petit was the first team to identify the genes that cause deafness. And they started to publish on gene therapy approaches. So we have at Sensorion the opportunity to exclusively have option rights on the program that comes from Pasteur, and our partnership has just been extended until 2028. Then it was very important to start working early with the medical care and the patient community, and that happened also since 2019 with the pediatric hospital Necker in Paris. And they started to do natural history studies, genotyping patients, following them up, and this natural history study was extended by Sensorion across Europe to identify otorphalin and GJB2. And those natural history studies, they paved the way to clinical trial because there was an early discussion with the medical community, and in our condition it means geneticist, audiologist, and ENT, but also discussion with patients and educating them on the prospect of gene therapy. And the last piece of the patient journey, it's the medtech player. And Sensorion has collaboration with first Cochlear. It's one of the leader of cochlear implants, and our study in the preservation of hearing loss after cochlear implantation is conducted with them. And also Sonova, it's a big market player of the hearing aids, and with them we're launching a natural history study on the early onset of early presbycusis. So this natural history studies that are starting early, it's a very important key differentiator to our competitors because it paves the way to the clinical trial and de-risk also our conduct of the clinical trial. And then all those partnerships, they feed our pipeline that I will go briefly through because we mentioned that already in introduction. So we have one small molecule that I will not develop today and 2 gene therapy. One, the first more advanced SENS501 is in otorphalin-related hearing loss. We're going to start the phase 1/2, and I will give you a little bit more details on the study design. And the other gene therapy is about GJB2. This is the biggest cause of congenital deafness, and it also corresponds to several populations, adult population, with early onset of presbycusis, or congenital at birth, or progressive during childhood. So now I will focus on those 2 first conditions and telling you a little bit that those 2 programs, they're coming from Pasteur and they're very rare. So otorphalin, the deletion in the gene leads to severe to profound deafness, and it accounts for about 8% of the cases of congenital hearing. Of course, for that, we have an orphan drug designation for the condition in EU and US, and we also have a rare pediatric disease designation in the US. On the other side, GJB2 hearing loss, as I said, it's the biggest cause of congenital hearing loss, but it also involves 3 populations. So the early onset at birth and progressive in childhood, it's about 200K patients. And in adult early onset of presbycusis, it adds about 100K patients. For those 2 conditions, there are no approved treatment. The standard of care is cochlear implantation, and the gene therapy will bring opportunity to restore a more physiological hearing. And that's what we're gonna start with our first study for SENS501. The study is called AudioGene. It has been approved through the EU new clinical trial regulation early in January with the decision in France. So it's a phase 1/2 clinical trial to assess safety, tolerability, and efficacy of SENS501 after unilateral injection into the cochlea for patients aged 6 to 31 months. So you might ask, why are they so young? This is because it's the best period to benefit from the treatment when there's enough neural plasticity to allow restoration of all the structure and allow language and speech acquisition. So the study will start with a dose escalation phase, three patients per dose, 2 doses, and will then continue to a dose expansions. In terms of endpoint, there is a safety and tolerability, and the efficacy endpoint is ABR for auditory brainstem response. Then I will move to our second product. It's about the GJB2 gene, and GJB2 encodes for a protein called connexin 26. It's a gap junction protein, and it's responsible for tissue homeostasis. In that gene, there are more than 100 recessive mutations that triggers deletion or truncation of the protein, and they all lead to hearing loss, severe deafness, and most are addressable via gene therapy replacement. And so on the right part of the slide, you can see internal data on the expression of the GGB2 protein, of the connexin 26 protein in the cochlea in mouse and non-human primates. And you can see in red that connexin 26 is expressed in different type of cells, and those are our target cellular population. And this is the same pattern in non-human primates and in human. And for that indication, what was maybe more a challenge was having adequate animal models, because connexin 26 deletion during development can lead to cellular degeneration. So full knockout model has a lot of limitation. And we are, with our partnership with Pasteur, we have access to adequate and unique animal models that are representative of our three population: deaf at birth, progressive, and early onset in adult population. Then, so here I will tell you a little bit more about the type of measure we have for efficacy. I talked previously about ABR. It means auditory brainstem response. What is it? It actually tells you how the cochlea is working with the brain pathways. And a normal person will have a threshold of expressed in decibel that is low, and when hearing loss comes, then the threshold in decibels increases. And if you had a hearing threshold at 120 decibels, it means you're completely deaf. So on the left part, the top graphs, it shows you a conditional knockout mouse model that leads to two phenotypes. You have in red, so the threshold is high, it's deafness at birth. And then in orange, it's a progressive hearing loss. So at 1 month, the phenotype is normal. And then from 2 months, and still you can see here at 6 months, it evolves into hearing loss. And on the bottom graph, you can see that our candidate is able to prevent the progression to hearing loss. So in blue, it's the injected ear with the candidate compared to the non-injected ear in orange. And on the right panel, you can see that this prevention of hearing loss correlates with the expression of the connexin 26 in the target cells of the ear, in green in the injected ear. I will now conclude with the status of our program. So we have 2 gene therapy programs, and maybe something I have not mentioned, it's AAV-based gene therapies. For otofeline, we're starting the first-in-man clinical trial, and we will be able to communicate safety data on patient by the end of the year. And then for GGB2, the IND-enabling studies are starting. We have 2 natural history studies ongoing on our 2 target population, pediatric and adult, and we foresee a clinical trial application in 2025. And I will be available after if you have any questions.